introduction. Hello, everyone. Welcome to today's webinar. I am Pardis Mohammadpour. I did my PhD in material engineering here at McMaster, and I joined CCEM in January 2023 as a postdoc researcher to work on our brand new X-ray imaging system. Okay, here is what I'm going to cover in today's webinar. I'll give a history of X-ray generation, which will be followed by an introduction to X-ray imaging. Then I'll talk about X-ray computed tomography or XCT. And then I'll introduce our new XCT systems in the CCEM. Afterwards, I'll discuss the data reconstruction, image processing, and XCT application. X-rays are heterogeneous electromagnetic waves with very short wavelengths in the scale of atoms. Since they have very short wavelengths, their energy is very high, so they can penetrate through the objects and reveal their internal feature, like this medical CT imaging you see below. I guess these images are familiar to all of us. Wilhelm Röntgen, a German mechanical engineer and physicist, discovered X-rays in 1895 and was awarded the first Nobel Prize in physics for that. Here you can see the first human X-ray image from his wife's hand. The X-rays started being used for medical purposes during World Wars I and World Wars II. The source of the X-rays can be natural, like cosmic radiation, or it can be man-made in the synchrotron or laboratory X-ray sources. Here I compared synchrotron and laboratory X-rays. The main differences is the source of the X-ray. In synchrotron, the electrons are accelerated to nearly the speed of light and forced to follow a circular or spiral path by strong magnetic fields. As they change direction, they emit synchrotron radiation, including X-rays. In the laboratory X-ray systems, X-rays are generated in a tube. Synchrotron is a very huge facility, costly to use, and usually accessible through proposal. However, lab-based X-ray systems are compact, cost-effective, and accessible anytime. The X-rays being generated in the synchrotron is intense and highly tunable, while in the lab, the X-ray isn't that intense, but the brightness is still reasonable for 3D imaging. I'll discuss the lab-based non-medical X-ray system further in the following slides because it's a focus of this presentation. Here I'm going to show you how the X-rays is generated in the lab. Here you can see the schematic of an X-ray tube. A cathode, usually a tungsten filament, emits electron and strikes an anode containing a target material. Electron excites the atoms in the target material, which release energy in the X-ray spectrum. There are two different types of emission from the atoms of the sample. The first one is the characteristic radiation, which is emitted when an inertial electron is ejected from an atom. An electron from a higher energy level fills the vacancy and it releases energy in the form of X-ray. The other emission is Bremsstrahlung, which means braking radiation in German, occurs when a high-speed electron is deflected or decelerated by the electric field of a nucleus, resulting in emission of X-ray radiation. Unlike the characteristic radiation, the energy spectrum of Bremsstrahlung radiation is continuous, ranging from 0 up to the energy of the incident electron. This continuous spectrum of X-rays produced through these Bremsstrahlung interactions provide the necessary information that we need for the X-ray imaging. Here you can see the schematic of an X-ray computed tomography or XCT system. It's basically a non-destructive 3D imaging technique. We don't need to cut or destroy the sample To be able to see the internal feature, we have an X-ray source, the detector, and the sample stage in between. The sample needs to be mounted on a proper sample holder and then loaded on a sample stage. X-rays are emitted from the source and penetrate through the sample. The differential absorption are picked up by the detector and shown as X-ray absorption contrast or 2D projection on the detector. Then the object is rotated and paused at different angles to collect 2D projections. After collecting several projections, the data is reconstructed using reconstruction software. Here is the 3D volume reconstructed data of this sample. And the next step is to use a 3D visualization software to analyze the data both quantitatively and qualitatively. Now it's time to introduce our new XCT systems here at the CCEM. The first one is the Zeiss X-Radia 630 Versa. This is the first installation of its kind worldwide. The highest spatial resolution is 450 nanometer, which is great for 3D imaging. The second one is the Nikon M2225. As you can see, it's a walk-in enclosure. It can be used to scan larger samples. The energy and power are higher in this system. I'll discuss each of these systems further in the following slide. Here you can see inside the X-Radia 630, the source energy varies from 30 to 160 kV. The metal target in this system is tungsten. There is a sample stage in the middle, and there are two different types of detectors, flat panel and objective detectors from 0.4x to 40x magnification. The flat panel detector contains 3072 by 1944 pixels. It's made of the scintillator, which converts the X-rays into visible light, which is combined with these photosensitive diodes to convert the light into the digital image. Detectors measure the intensity of the X-ray. Intensity is essentially the quantity of the X-ray per pixel per unit of time. Transmission, or the ability of the X-rays to pass through the sample, is another important concept in XCT. When the material is denser or thicker, the X-ray absorption is higher, and subsequently the transmission, the intensity, and the brightness are lower. That's why it appears darker on the image. With flat panel detector, we only have geometric magnification. There is a cone beam in this lab-based X-ray system. So based on the similar triangles method, the magnification can be calculated as the distance between the source and detector divided by the source and object distance. The objective detectors are a combination of scintillators, again, to convert the X-ray into the visible light, combined with high-resolution detector optics and CCD detector capturing the visible light image. The detector has 24 by 2024 pixels at the maximum binning in this system. Here we have two means of magnification, and the total magnification is product of optical and geometric magnification. By combining these two magnification, the system can achieve a resolution performance of 450 nanometer across the full range of energy from 30 to 160 kV. There are also 13 different source filters and 3 DCT apertures mounted on the source wheel. This wheel can rotate and enables us to apply these filters on the X-ray path. But why we apply filter on the X-ray beam? Filters are usually made of pure metal sheets with different thicknesses, and they are applied between the source and the sample, as you see here. Now let's see how it works. For example, when the electron accelerated voltage is 60 kV, then the energy spectrum of X-ray radiation is continuous, as discussed in the previous slide, ranging from 0 to 60 keV. The red box indicates the low energy photons needed to obtain contrast in low Z materials. After applying the filter, the low energy photons are absorbed by the filter. So it alters the spectrum, and the spectrum shifts towards the high energy portion significantly, as you see in this green box. When the filter is thicker or made of higher density material, it moves the spectrum even more towards the high energy portion. This is beneficial for imaging high Z materials. We generally apply filter to increase the penetration or remove low energy X-rays, especially when we do high energy scanning and we need more uniform high energy beam. You see in this example that the image brightness has improved after applying the filter significantly. The second reason is to reduce beam hardening artifact. When the X-ray beam passes through an object, different materials or similar materials with different thicknesses attenuate X-rays to a varying degrees. This makes the X-ray beam heterogeneous. That creates this beam hardening artifact. For example, in this example, the outer layer looks brighter, but after applying filter, we helped making the beam more uniform. And the third reason is to reduce the scatter at the detector. When we scan high density materials, some of the lower energy X-rays are scattered and create scattering artifact, as you see in this image. However, by applying filter, we can remove this scattering artifact. The user interface in X-Radia 630 is called NAVEX. NAVEX is only available on 630 version. The previous versions use Scout and Scan software. The very first step in X-ray imaging is targeting the region of interest, or ROI. In the NAVEX system, there are two ways to scout the ROI. One is the projection scout, and the other one is the volume scout. The first step would be taking a single 2D projection image to decide which scout method is proper for the sample. For example, if you want to do a high resolution imaging on the iPhone's camera, we can easily target it on a single radiograph. So it's reasonable to use the projection scout. However, to target a specific feature in this rock, we cannot do it based on the 2D projection view because we cannot basically see any features. In this case, we use the volume scout tool, in which we do a very quick overview scan and reconstruction, and then we can pinpoint our volume of interest for high resolution imaging. The other new tool that NAVEX presents is the guidance workflow. The guidance is directly embedded in the software. You must take a single 2D radiograph from your sample and identify the target region. For example, black box is our target region in this sample. Then the system gathers some information on the transmission and intensity at the identified region, then optimizes the parameters and provides you with some recommendation on the parameters. It also provides some images that's showing the effect of different parameters on the image quality. It can be this guidance workflow actually can be very useful for new users to become independent faster, and it can also reduce the time that we need to set up the scan significantly. Phase contrast is one of the techniques that can be applied in our X-Radia system to enhance the phase contrast between phases in low Z materials like biological or polymer specimens. Its most advantages on smaller samples, since these high magnification objectives can be used to enhance the quality of phase contrast and limit the possible noises. It's a propagation-based phase contrast, in which by expanding the distance between the source and a detector, we can increase the image contrast by highlighting small details of these differing refractive indices within a structure that would be uniform in the short distances, as you see here. And here you can see two radiographs from a mouse knee. which were taken with the source and detector distance of 22 millimeter and 110 millimeter. As you can see, the tissue between the knees look uniform at short distances. However, by increasing the distance between the source and detector, we could differentiate between the tissues, the different tissues available in this structure. After the data reconstruction, we need to data acquisition. We need to reconstruct the data. The data reconstruction involves combining the 2D projections using mathematical algorithms and generating a detailed 3D image of the object. The most common reconstruction methods are filtered back projections and FDK. Other than this standard reconstruction algorithm, Zeiss Reconstructor software offers a deep learning-based reconstruction toolbox called DeepRecon. It enables fewer projections for sample classes with repetitive workflows, which this reduces the scan time significantly. There is also another image quality model in which we train the model based on the CT data, and it will increase the image quality for that data set by reducing the noise in the image. Here is an example of a 2D reconstructed cross-section using standard recon and a deep recon. As you can see, the image quality is enhanced significantly in this eutectic high entropy alloy after applying the deep learning model. Lab DCT for diffraction contrast tomography available exclusively on the X-Radia 630 and 620 Versa. This enables non-destructive mapping of grains in 3D. It's developed by Zeiss collaboration partner Exnovotech. This technique is very useful in characterization of polycrystalline materials like metal alloys and geomaterials. LabDCT offers specifically information on grain centroid position, grain size, grain orientation, shape, and grain boundary information. There are two detectors that can be used for DCT to acquire high-resolution crystallographic information. We can use the 4x DCT objective, and for a larger sample in which we want to map a larger area, we can use a flat panel detector. There are two other other elements for DCT. The first one is beam apertures, which constrain the incoming beam. They're mounted on the filter wheel, and we can switch automatically from between them. And the second element is the beam stop, which blocks the direct transmission beam, and they are mounted on the DCT-4X and flat panel detectors. There is still some limitation with the lab DCT technique. For example, the sample should be strain-free, and there is grain size limitation. The size of the grains has to be between 50 to 300 micrometer to be optimal for the DCT, and twins are counted as individual grains. DCT works based on the law diffraction, in which set of planes within individual grains fulfilling Bragg's condition generate diffraction spots on the detector, as you see here, and eventually the raw DCT data looks like this, which is called a low diffraction pattern. The next step would be segmentation and indexing the pattern using the Grain Mapper 3D software. This software is a 3D crystallographic grain reconstruction software developed by Exnovotech, the same company that made the whole DCT software. And here is an example of reconstructed DCT data on the pure iron. As you can see, we have the 3D information on the crystallographic orientation. Now I'm going to talk more about our Nikon XCT system. There are three different X-ray sources in this system with different characteristics suitable for different applications. Depending on the size of the sample and the required resolution, we can switch from one source to another. These sources are called rotating target, static target, and transmission. In the rotating target, the metal target is tungsten, and the maximum voltage is 225 kV. The tungsten target rotates and helps avoid the local heat on the target, enabling us to have a higher power up to 450 watts. This higher wattage is very useful for scanning very large samples. This source is suitable for imaging parts that requires pixel size larger than 15 micrometer. In static target, the metal target is static, which limits the voltage. The maximum voltage in this source is also 225 kV. There are four different metal targets are implemented in the static target source, tungsten, molybdenum, silver, and copper. Different target materials produce different X-ray energy spectra as discussed in the previous slides, making them suitable for specific application. A static target can be used for samples that requires pixel size between 5 and 15 micron. And the third source is the transmission targets. This source has a smaller spot size and allows a higher magnification as sample can be positioned much closer to the target. The resolution can be as good as 3 micron using this target. When the required pixel size is smaller than 5 micrometer, the transmission target is the best option to use. But if we need higher resolution than 3 micron, our Zeiss X-Radia system would be a better choice. Unlike the Zeiss system, only a flat panel detector is available in this Nikon XCT, which means that we only have geometric magnification. The flat panel can move vertically and horizontally. That enables us to scan a very large field of view, 50 centimeter by 50 centimeter. The sample stage can carry up to 180 kilogram weight. That enables a scan of large samples and in situ experiment accessories. Here are two examples of large samples that we have scanned with our Nikon system. The first one is a frozen human arm. The sample was very large and had to stay frozen during the experiment. We didn't even need to take it out of this plastic cooler to scan the sample, so the temperature was controlled during the imaging. The other sample is this ancient pot. Studying the archaeological sample has to be non-destructive. This ancient pot was too large to be characterized by any other characterization techniques. The Nikon XCT system was used to analyze the design and microstructure of the specimen without damaging the sample. Silotech micro-tensile stage is one of the in situ equipment that we use in the Nikon system. The load capacity in this stage is 1.6 kilonewton. Performing in situ tensile tests in the XCT systems involves combining the capability of XCT with the mechanical testing to observe the internal deformation or behavior of the material under the tensile or compression forces. We will install another micro tensile stage with a higher load capacity up to 10 kilonewton in March 2024. This new stage is shorter and can be fit in both systems. The next development in our in situ mechanical testing setup would be to equip the tensile stage with a furnace to be able to do high temperature mechanical testing and tomography. 3D imaging techniques such as XCT are meaningless without data analysis. Visualization software plays a crucial role in the post-processing and analysis of 3D imaging data. We use Dragonfly software for the 2D and 3D visualization and image processing, and this software provides interactive tools that enable users to manipulate and navigate through the data, zooming in on a specific region of interest, rotating the data, and adjusting transparency and color mapping for better understanding. And furthermore, it allows users to create cross-sectional views of the 3D data, as you see in this picture. And this is very useful for examining internal structure layer by layer, because we can move through the layers, as you see in this video. The visualization software also facilitates the segmentation of structure within the 3D dataset. This process involves isolating specific regions of interest, which is crucial for quantitative analysis. For example, in this sample, we segmented the pores in this high entropy alloy. Then we can measure and analyze the properties such as volume, density, pore fraction, or other properties within this segmented region. With all being said about the XCT, it has application. It has application across various industries and materials. Here are some of the key sectors and materials where XCT is commonly used. In manufacturing, aerospace, and automotive industry for quality control and to inspect the and verify the integrity of the critical components. XCT can also be used for to examine items such as weapons, explosive devices, or other objects relevant to forensic investigation, because it provides detailed 3D images of the internal structures without physically altering the evidence. And it can also be used in geology and earth sciences to study the internal structure of the rocks and soil, to understand the composition and characterization of the geological formation. In R&D sectors, XCT is utilized in various scientific disciplines like physics, chemistry, biology, and engineering for non-destructive 3D analysis. It can be used in electronics and battery industry or research to inspect the internal structure without disassembling the part. It can be used in life sciences for animals and plants, for example, visualizing and studying the anatomy of small animals like mice and rats to understand disease models and treatment effects. And also in paleontology and archaeology researches, we can examine fossils without damaging them and study the internal composition of artifacts, which provides insight into their construction and the historical content. The other application of XCT is the correlative microscopy. By combining our capabilities here at the CCEM, we can create a novel pathways and methodologies for multi-scale, modal analysis. This equipment allows us to study the materials over a wide length scale range from centimeter to atomic size. We can target the region of interest non-destructively using the XCT systems, then the sample preparation using the FIB-SEM laser, and characterization of the microstructure using our other high resolution microscope. And at the end, I would like to thank you all for attending this webinar, and I hope you enjoy the webinar, and I would be happy to answer your questions if there is any.
Great, thank you, Pardis. If anyone has any questions, please put it in the Q&A. We'll just wait 20 seconds or so to see if any questions pop up. Please remember that these will be posted on our YouTube channel, so if at any point you'd like to rewatch the video, it will be available. And if you have any interest in using any of the instruments here at the center, including the systems Pardis showed today, they are all set up on our equipment training modules, so training is available for all our instruments here. Just feel free to reach out to myself or to Pardis, and we can get you set up in your training and using our instruments.